Hi, I'm Susie Spriggs. I'm the tutor manager for the sciences at the ALC, and I tutor various courses including biology, chemistry, calculus, and psychology. For this mock session, I'll be the tutor. Enjoy!
Hi, my name is Leticia Borges, but you can call me Letty. I am a senior tutor at the Academic Learning Center. I am also a science tutor, so I tutor Bio 151, 152, Chemistry 105 and 106, and Math 131. I am going to be the tutee in this mock session. Hope you guys like it.
Hi, Leticia. How was your week?
Hi, Susie. My week was good. Bio was a little hard. We just started talking about kidney function, osmoregulation, and water concentrations, and I was really confused on that topic.
Okay. Would you like to go over some of that today?
Yes, I really wanted to go over one of those topics because we're gonna have an exam next week on it, and I feel really nervous about it because I feel like it's not my strong forte.
Okay. So do you understand like the basic kidney structure, or is it more the nephron stuff that you're having trouble with, or are you having trouble with all of it?
So I understand that the kidney is basically like a filtering system, and it has all these tubes that connect to different concentrations. But I'm just confused on where everything is transporting, like on what time, and also the labeling of the nephrons, kidney little nephrons. I also am confused on that.
Okay. So do you happen to know what any of those little tubes that you're referencing are called?
No, I forgot the name.
Okay. So here, wait, I'm going to draw a picture real quick and see if you can like pick out anything, what it might be.
Okay, that's awesome because I am a visual learner.
Okay. Hold on. Sorry about that.
You're good. You're good.
Okay. Okay. So do you recognize like the shape of that at all?
A little bit. I recognize that the middle part where it dips really low, that's like, oh, I forgot the name. But I know that the red part is where it starts filtering out. And then it goes from the red part all the way down. And then the last part right there, the green part, that's where it goes down to the urethra and exit out as urine. That's basically what I know.
Yep. So the red part is called the glomerulus. And what did you say that did again?
I think it uses like osmoregulation to like filter out salts, but I'm not sure.
Yep. And then so that's where your blood and gets filtered out and it gets the plasma taken out. So the glomerulus is a capillary bed. So all your blood comes in one side, it filters out, all the liquid stuff goes into your nephron, and then everything else goes back into circulation.
Oh, I didn't know that. I actually thought that the blood actually went through the whole green part too.
Nope, just the plasma or the liquid part.
Okay, cool.
So can you point out where a Bowman's capsule might be?
A Bowman's capsule. Is it the thing that's surrounding the glomerulus?
Yep, and what does that do?
It collects the plasma coming out of the glomerulus and makes it travel down the other tubes, I believe.
Yep. So then you also have your proximal and your distal convoluted tubule. So can you point those out?
Okay. I are.
So if you just had to take a random guess, which do you think they might be on here?
The proximal one, I think it's the one near the glomerulus.
Yep.
And what was the name of the other one that you said?
Distal convoluted tubule.
Is that the one that comes right after the proximal one?
So like the squiggly part or the big loop?
The squiggly part right after before the big loop.
Yep, so this side is your distal one, and over here is your proximal one. Because
Oh,
proximal means close, so proximal is close to the glomerulus, and distal is far away, so it's far away from your glomerulus.
oh, that helps me because I was confused. Could you point to where you're naming this stuff, because I was confused where the proximal and distal were.
So there, our glomerulus is here as we saw, and then here would be our Bowman's capsule. So then this one closest to your Bowman's capsule or glomerulus is going to be your proximal convoluted tubule because it's close. And then this tubule over here that's convoluted or really, really squiggly is going to be your distal one because it's further away.
Okay. What's the big loop in the middle?
So the big loop in your middle is called your loop of Henle.
Loop of Henle. I remember that I was looking at a diagram in class and I was really confused because on one side of it, the salts were coming like in, and the other side of it, the salts was coming out. So can you like point to me where the movement of the salts are going?
Yep. So first off, which would be like your descending loop of Henle, and which one's your ascending loop of Henle?
Oh, that one I know. The descending one is going down and the ascending is going up.
Yep. So your descending will be over here and your ascending will be over here.
Yes.
So then on your descending one, either I'll give you two options and you pick one and see if you can pick which one is right. So either descending, your water's going out or your salt's going out.
I think the salts are going out.
It's the water.
Really? Why?
Yeah. I don't really know if there's a particular reason why, but one way to remember it is down at the bottom is where it's going to be the most concentrated or the most like salts and stuff left. So the only way to get it the most salts down here is the water has to come out first.
Okay. Just give me a second while I take some notes.
Okay.
Okay, so descending loop, the salt stay, the water comes out, and the most concentrated at the bottom. Okay, thank you.
Yeah, so then what do you think happens on the ascending loop?
The water goes in and salts come out.
So it's only the salts come out. So the water is only permeable, or it can move through on this side, and on this side only salt can move through.
Is that because there's gated channels that control the movement of water on one side and the movement of salts on the other side?
Yep.
Oh, okay.
And then do you know what this long tube over here is called?
Give me 1 second now. Someone's knocking on the door.
Okay.
Never mind, my roommate got it. I don't remember the name, but I know that that's the one that connects to I think the urethra? Not sure.
Yep, it eventually leads into the urethra and it's called the collecting duct.
The collecting duct, okay.
Yep. And then is it just one nephron that leads into a collecting duct, or are there going to be multiple nephrons leading into a collecting duct?
Multiple, because that section is just a little small section of the nephron. And there's many nephrons in a kidney.
Yep. So do you want to try explaining the process back to me?
Yes. So the red part is the glomerulus, and that's where the blood gets filtered out, where the plasma gets taken out. And then the thing that looks like a hook, oh, the name. I know what it does. It collects the plasma. It's called the fisherman's hook. I forget. But I know what its function. I just need to look over the names. The squiggly line right next to it, that's the proximal, what was the name again? Proximal, I forget. No, seriously, I forget. What was it? Proximal
Convoluted tubule.
There you go. Proximal convoluted tubule. That over there is right next to the glomerulus. Well, next to the hook, which is next to the glomerulus. And that's why it's called proximal. And then there's the descending loop of Henle, and then the ascending loop of Henle, which is just altogether called loop of Henle. And then next to it is the distal convoluted. Oh my God, I really said the name. Distal convoluted. The proximal convoluted. I forget.
Sounds like tube.
Tube.
Yep.
So distal and proximal convoluted tubes, right?
Yep.
Okay, so glomerulus, the hook of Henle, right?
Yep. Loop of Henle is this there. Yeah.
No, the one next to the glomerulus. It's
That's
called
your Bowman's capsule.
Bowman's capsule. There you go. So the glomerulus, the Bowman's capsule. Then there's the convoluted, the proximal convoluted tubes. Then there's the loop of Henle, which has the descending and the ascending. And then we have the distal convoluted tubules. And then we have I know this one. Don't tell me. The catching? No. Oh my God.
It's where all the nephrons go and collect together.
Collecting duct. That's what I was saying. There you go.
Okay. So out of your ascending and descending loop, what do they do?
So the descending loop takes out the plasma or the water on the solution, and it's most concentrated at the bottom. And then going upward, the ascending is where they take out the salts.
Yep.
And then that is just they collect the urine.
Yeah. And then what's fancy about the collecting duct? What can be like added or taken away from the collecting duct?
Either salt or water can be added or removed depending on the concentration.
So actually in the collecting duct, since you're already almost at the end, you can't really take back any more salt at that point. At that point, whatever salt's left is going to be excreted out of you. But what else can be taken out or added?
Water.
Yep. And what are like the fancy name of the little channels that they can go through?
Aquaporins.
Yeah. So what happens if you add more aquaporins or take away aquaporins?
So if you have more aquaporins, more water is going to diffuse into the collecting duct. But if you take away, less water is going to diffuse out or into the collecting duct. Right?
I think you have it backwards, because if you have more aquaporins, you're going to be drawing more water back into your body. But if you take away aquaporins, you're going to be letting more water out of your body.
Oh, is that because of osmotic regulation due to concentration compared to the outside of the body? So if you have less aquaporins, less water will be able to move out?
Yeah.
Okay, yes, I always am confused about osmotic regulation, which is why I have a question about it too.
Okay. Is there anything else about the nephron or the kidney you would like to go over?
I think I got a pretty solid handle on it after you explained it. Now I just really wanted to go over osmoregulation, especially because, talking about the collecting duct, I got confused between aquaporins. So I was wondering if we could go over that.
Is there anything specific that you want to go over with that?
So I'm always really confused on how to determine like hypertonic, hypotonic, and isotonic solutions. Where the concentration will move and why it moves that way.
Okay, so do you know what either the words hypertonic, hypotonic, or isotonic mean, or if you don't, what their roots mean?
So I kind of have like a little way to remember it. So hypo, I think of like hippo, and hippos are fat. So I feel like water moves into a hypotonic solution. And then a hypertonic is just the opposite of hypo, water moves out. And then isotonic is like the middle. It's like a net movement of concentrations going in and out that doesn't change.
Yeah, that's one way to remember it. Another way is that hyper means more, so it's going to have more of your solute. So when you have more of the solute, the water is going to end up moving. Do I have that backwards? No. Hold on. I'm going to draw out what I'm trying to say.
That helps me too.
Okay. So if this is like our inside of the container, and out here is our outside of the container, or technically semi-permeable membrane, so it can actually move in between. And this is inside. And these little purple dots are your solute or like your salt or something. And water's just everywhere else, and we can think about that. So inside the container or membrane, how many things of solute do you see?
5.
Yeah, and on the outside there's how many?
2.
Yeah, so would there be more water on the outside or the inside?
Outside?
Yeah, so in terms of water, since can solute move between the membranes normally?
No.
No, only water is the only thing that can move osmosis or naturally without any pores.
Oh, okay. Let me write that down.
Okay.
Okay.
So then if we said water would move in, what would happen to this cell or membrane on the inside? Would it burst, get tiny, or stay the same size?
It would burst, so it would be hypotonic.
So you can think of, would the solution be hypotonic or would the cell be hypotonic? If we're looking at just this picture here.
The cell will be hypotonic because the concentration is going into the cell, like the water is going into the cell.
In this case, our solution would be hypotonic because hypo means less, so it has less solute, more water, and in here our cell would be hypertonic because it has more solute, less water.
Oh, I always got confused on that because I always thought it was talking about the movement of water. So if the water moves into the cell, that means that the cell is hypotonic.
Yeah.
So, but in terms of solution, the outside solution would be hypotonic because it has less products because hypo means less.
Yeah.
But the inside solution inside the cell will be hypertonic.
Yeah. So in thinking of like your hippo explanation, you know that since your cell ended up getting bigger, it would end up being in a hypotonic solution because the cell itself got bigger.
Which means that it has more water or the same amount of water compared to solutes.
Yeah.
Oh, okay. Got it.
Okay. So wait, if we take away those, what would of hypertonic, hypotonic, and isotonic, what would you call this?
Isotonic?
Yep, and how do you know it would be isotonic?
Because there's the same number of solutes inside the cell and the same number of solids outside the cell, so there's not going to be more water going in or out the cell because it's the same number of solutes.
Yep, and then if we add so now what would your inside and your outside be?
The inside, okay, I have to have like a mental walkthrough.
Okay.
So there is less solutes inside.
Yeah.
More solids outside, which means that water is going to move to the outside because there's more solids outside. So that means that the solution inside the water, inside the cell is hypertonic because it has less solutes.
The solution or the cell is hypotonic?
I was going to say that the cell was hypotonic because it has less solutes.
Yep.
And then the outside is hypertonic because it has more solutes.
Yep. And then what would happen to your cell? Would it grow? Would it shrink? Would it stay the same size?
It would shrink because it would be losing water.
Yep.
Okay. Could we also talk about the names of, I know there's like special names of how plants and cells and animal cells in different solutions, like when they shrivel or stuff like that.
Oh, like the fancy names for them?
Mm-hmm.
So when they shrivel, that is called, hold on, let me make sure I don't have it backwards. Okay, so when they get smaller, or when they shrink rather, that's a cell lysine, which just means it's going to end up shriveling up and getting tiny and eventually bursting just because of how much it's shriveling up.
That's just animal cells, or that's plant cells too?
That's just animal cells, because what do plant cells have that animal cells don't?
A cell wall
Yeah,
and a vacuole.
yeah. So their cell wall is what makes their cell will still shrink, but when you think of an animal cell, it's going to shrink up really tiny like a raisin. But an animal cell would be like a cube, so you would just shrink up what's inside, but it's still going to stay a cube shape.
Oh, okay. All right. So what would the name for a plant cell in that state be? Or is there no name?
There's no really specific name for that one.
Okay. It would just be shrivel.
Yeah.
All right. What about when they burst?
So when they burst, that's when they lyse open from bursting, or it's called turgid, and they just get big.
And that's for animal cells?
Yeah.
Okay. And what about plant cells?
Plant cells, I don't think there's really any specific name either.
Okay. And what about if they're isotonic? Do we just say isotonic, or is there just another?
Yeah, just isotonic, normal.
Okay. All right. Thank you. This really helped
Yep.
me. Those are all the questions I have for today.
Okay. Is there anything that you want to go back over, or do you think you understand most of today well?
Is there a way for you to send me pictures of the drawings that you did, just so I can visualize them better? I try to draw them on my notebook, but I have a difficult time following you through video. So it would really help me if you could send me pictures, if you can.
Yep, I'll write them down and email them to you after the meeting.
Okay, thank you. That would be really helpful.
Okay. So for this week, do you want your goals to be, or what do you want to mainly work on this week?
I want to really hammer down all the type of solutions and concentrations, especially osmoregulations and kidney functions, because we're doing kidney right now, and I really need to really know where the concentrations are moving and the salts and moving throughout the whole nephron. Because we're going to have an exam next week on Wednesday, and it's going to be an exam. So I want to get that down, and I also want to be able to remember the names, because I have a really hard time remembering the names of each part. And there's a lot of little different parts in the nephron, and not just the nephron, in every section in bio. So I want to really memorize all the names and parts.
Okay. And then since you have a test coming up, do you want to meet anymore, or do you think you feel okay for your test coming up?
I think I need to study a little bit more, and figure out what's my strong suits and weak suits in material wise. And then if I do have any questions, or if I do want to meet up, can I email you later in a week and see if we have some free time?
Yep, that works.
Okay.
And don't forget to, if you have any questions specifically for your professor too, you can stop by their office hours.
All right. Thank you for letting me know.
Yep.
All right.
Okay. Have a good rest of your week.
Oh, and don't forget about the logging.
Oh yeah. Sorry, I completely forgot about that too.
You're good,
Okay. So I'll go ahead and bring up my tutor log now. Thank you for reminding me.
Yeah, don't worry. I know. It's okay. It happens to the best of us.
Yeah. Okay. So for today, I'm recording us from 8:15 to 8:45, which is a half an hour. And then I'm writing down that I was tutoring you. Does that sound okay?
Yes, that does. All right.
Okay.
All right. Thank you for your help. Have a good day.
You too.
Bye-bye.
Bye.